A recent study published in the journal Nature Aging is exploring a novel approach to treat neurodegenerative diseases like Huntington's by drawing inspiration from an unexpected source, the trunks of trees. The study's primary objective was to combat the protein clumping and associated symptoms of Huntington's disease, a neurodegenerative disorder classified as a proteinopathy. Huntington's is one of 9 such disorders characterized by the accumulation of toxic protein aggregations called polyglutamine, which do not naturally adhere in healthy human bodies. In both humans and animals, this condition leads to the demise or malfunction of brain cells. Surprisingly, plants also experience proteinopathies akin to Huntington's, but without any detrimental consequences. Dr. Ernesto Lamas, the lead author of the study from the University of Cologne's CECAD Cluster of Excellence for Aging Research, initiated a remarkable experiment to unravel how plants manage these toxic protein accumulations. He introduced the toxic mutant protein, Huntington, known to cause cell death in human neurons, into plants, specifically Tale cress. Contrary to animal and human models, Dr. Lamas discovered that Tale cress plants actively removed Huntington protein clumps and remained unharmed by their presence. Seeking to determine if this effect could be replicated in animals, the research team delved deeper and pinpointed the chloroplasts, plant-specific organelles responsible for photosynthesis, as the key reason why plants are immune to the adverse effects of mutant Huntington protein. Unlike humans, Dr. Lamas explained, plants have chloroplasts, an extracellular type of organelle that could provide an expanded molecular machinery to get rid of toxic protein aggregates. The research team identified a specific chloroplast plant protein called SPP as the underlying mechanism responsible for the resilience of plants to the problematic human protein. They further observed that introducing the plant SPP into models of Huntington's disease, including cultured human cells and nematode worms like C. elegans, resulted in a reduction of protein clumps and alleviation of disease symptoms. Dr. Hai and Ju Li, a postdoctoral researcher involved in the study, expressed their satisfaction, stating, "We were pleased to observe that expression of the plant SPP protein improved motility of C. elegans worms affected by Huntington even at later aging stages where the symptoms are even worse." The findings of Dr. Lamas' study hold great promise as they open up the possibility of investigating SPP as a potential therapy for Huntington's disease. Furthermore, it encourages the notion that plants may play an unexpected role in advancing treatments for human diseases. Dr. Lamas highlighted the resilience of certain plants, noting, "Many people don't notice that plants can persist amongst variable and extreme environmental conditions that cause protein aggregation. We usually forget that some plants can live thousands of years and should be studied as models of aging research." This discovery is not merely theoretical, it has practical implications. Dr. Lamas and his team believe there is tangible value in their findings. Study co-author Dr. Seda Koyanchu has announced their intention to establish a startup focused on producing therapeutic proteins derived from plants and testing them as potential treatments for neurodegenerative diseases in humans. This innovative approach could potentially offer new hope for individuals affected by these devastating conditions.